brain is very complicated, and there's so much we still don't understand. Like, how do diseases like Huntington's affect the brain? Huntington's disease is a rare but genetically fatal disorder that affects the brain and causes physical issues like involuntary muscle movements and difficulty speaking and swallowing. It also causes mental issues like difficulty learning new information. It's like having ALS, Alzheimer's, and Parkinson's all at once. To get this disease, we first take mom, who in this case has a normal Huntingtin gene and no disease, and dad, who has one mutant Huntingtin gene on his chromosome. If they have 4 kids, then by probability, 2 will have the disease and the other 2 won't. This is called an autosomal dominant disorder, which means they can have 1 copy of the mutant gene and still get it. When looking at dad's chromosome up close, we'll focus on the Huntingtin gene specifically. What makes it different from the norm is that the CAG trinucleotide repeats are greater than 35. This in turn creates a Huntingtin protein that has the same mistake translated into its structure, which leads to neuron death. Although we don't know the specific function of this protein, we know that individuals with this altered gene have lower BDNF, or brain-derived neurotrophic factor levels, which are proteins that help with neuron growth and survival. This altered gene also leads to protein aggregates, which damage the neuron as well. Looking at how Huntingtin causes aggregates, first we see that when the protein is translated, a cell signal for proteases like caspase 3 and 6 comes, and these proteins cut the Huntingtin protein down to be digested by the cell. But some of the fragments are toxic and can lead to protein bundling around the axons and dendrites of a neuron, called neuronal inclusions, leading to damaged neurons. When some of these fragments enter the nucleus, they interact with proteins like CBP or CREB binding protein, which is regulatory of DNA and key for cell survival. The glutamines on the CBP interact with those on the mutant Huntingtin protein, preventing them from functioning and forming more aggregates. This leads to fewer CBP being produced, which leads to nerve cell death. Another mechanism that forms aggregates is through the ubiquitin proteasome system, or UPS. When the protein gets tagged with ubiquitin, a proteasome comes to degrade it, but it gets inhibited by the altered protein for unknown reasons, and the altered gene continues to produce proteins that attract proteasomes and keep forming these aggregates in the cell, which forms this ever-going cycle leading to the damage seen with this disease. So how do we stop this? One way is through gene therapy, which is very beneficial because it lasts for years. Through adeno-associated virus, or AAV, DNA for a gene that codes for RNA is implanted to the cell to counteract the mutant Huntingtin protein, leading to less of it, which means less toxicity and aggregates, which is an important step in addressing this disease. Another important thing is that when the AAV delivers to the striatum, the area most affected by Huntingtin, some therapies have shown signs of spreading to the frontal lobe area, which is a great sign. Hopefully we find a cure. Thanks for watching.